The two main efforts of antiviral defense are prevention of viral infections by vaccines and treatment of viral diseases with drugs. What we're going to talk about in this lecture will be vaccines, how they work, how they've been developed, and then in the next lecture we'll look at some of the drug treatments that are available. When a disease stops circulating in a region, it's considered eliminated. Polio, for example, was eliminated in the United States by 1979 after widespread vaccination efforts. If a particular disease is eliminated worldwide, it's considered eradicated. To date, only one infectious disease that infects humans has been eradicated. In 1980, after decades of efforts by the World Health Organization, the World Health Assembly endorsed a statement declaring smallpox was eradicated. Coordinated efforts rid the world of a disease that had once killed up to 35% of its victims and left others scarred or blind. Smallpox eradication was accomplished with a combination of focused surveillance, that is, quickly identifying new smallpox cases, and ring vaccination. Ring vaccination meant that anyone who could have been exposed to a smallpox patient was tracked down and vaccinated as quickly as possible, so effectively corralling the disease and preventing its further spread. The last case of wild smallpox occurred in Somalia in 1977. Smallpox was a good candidate for eradication for several reasons. First, the disease is highly visible. Smallpox patients develop a rash that is easily recognized. In addition, the time from exposure to the initial appearance of symptoms is fairly short, so that the disease usually can't spread very far before it's noticed. Workers from the World Health Organization found smallpox patients in outlying areas by displaying pictures of people with the smallpox rash and asking if anyone nearby had a similar rash. Second, only humans can transmit and catch smallpox. Some diseases, in contrast, have an animal reservoir, which means they can infect other species besides humans. Yellow fever, for example, infects humans, but can also infect monkeys. If a mosquito capable of spreading yellow fever bites an infected monkey, the mosquito can then give the disease to humans. So even if the entire population of the planet could somehow be vaccinated against yellow fever, its eradication could not be guaranteed. The disease could still be circulating amongst monkeys, and it could reemerge if human immunity ever waned. The discovery of an animal reservoir for yellow fever was, in fact, what derailed a yellow fever eradication effort in the early 1900s. Smallpox, however, can infect only humans. In effect, aside from the human population, the virus has nowhere to hide. Equally important is the ability to protect individuals against infection. People who survive smallpox naturally developed lifelong immunity against future infection. For everyone else, vaccination was highly effective. The WHO trained vaccinators quickly, and they could immunize large groups of people in a short time. The eradication of smallpox raised hopes that the same could be accomplished for other diseases, with many named as possibilities, for instance, measles, mumps, and polio. So let's talk about what's been happening with polio. In the first half of the 20th century, polio epidemics appeared each summer in the U.S. At its peak in the 1940s and 1950s, polio would paralyze or kill over 500,000 people worldwide every year. The fear and the collective response to these epidemics would give rise to extraordinary public reaction and mobilization, spurring the development of new methods to prevent and treat the disease. In a press conference in April 1955, it was announced that the Salk polio vaccine trial was a success. The vaccine, they said, was 80 to 90% effective against paralytic polio. The U.S. government licensed Salk's vaccine later that very same day. The press conference and the licensure paved the way for widespread distribution and use of the vaccine. Just a few weeks after the landmark press conference announcing success of the vaccine trials, an Idaho doctor reported a case of paralytic polio in a recently vaccinated girl. Over the next few weeks, similar reports trickled in to local health authorities. All involved a disturbing detail. The paralysis in these children began in the vaccinated arm rather than in the legs, which was more common in a natural infection. It soon emerged that most of the cases of paralytic polio occurred in children inoculated with vaccine produced by the Cutter Laboratories in California. A subcommittee of researchers and public health officials made several changes to polio vaccine production methods in order to address the problems that became evident from the Cutter disaster. They required manufacturers to test larger samples of each vaccine lot, to lengthen the time the vaccine was treated with formaldehyde, to use filters that would remove clumps of virus that might resist chemical inactivation, and to test the vaccine after bottling, amongst other changes. The vaccine program resumed, though some locations declined to participate because of potential risks from the vaccine. The U.S. Surgeon General then suspended the polio vaccine program in order to investigate the safety of all 6 manufacturers' vaccines. The full review concluded that 11 people died from the vaccine and hundreds were paralyzed. Though the cause of the disaster was never proven, it is likely that certain production methods, which, it turns out, did not follow Salk's instructions, resulted in a failure to completely kill the type 1 polio in the vaccine. Another controversy was when Merck researchers detected a simian virus in monkey kidney cells used to grow poliovirus for Merck's polio vaccine. They later showed that the simian virus, SV40, caused tumors in hamsters. Merck eventually withdrew its polio vaccine from the market. By 1963, government screening programs began to look for simian virus in polio vaccines. In 1960, the link between viruses and cancers was a new idea, and it was suggested that all Salk-type polio vaccines be withdrawn until the problem could be addressed. Today, polio vaccines are routinely screened for all known viruses. Many studies have been done to investigate if there have been any adverse effects from the SV40 in the early polio vaccines, and none have shown a relationship between SV40 and cancer in humans. So the people who received that early vaccine with the contaminant did not suffer any ill effects. The SV40 finding, though, was responsible for a shift away from using monkeys for tissue culture. Researchers would largely use human cells for the next wave of vaccine development. Another problem with the polio vaccine was an unfounded rumor that the polio vaccine had contained HIV, and that's how HIV was introduced into Africa. Molecular studies and the discovery of people who had HIV long before any polio vaccine program started debunked this, but still, you all have firsthand experience about how these rumors can get started and how hard it can be to overcome them. And then lastly, distrust of government has caused problems with people participating in polio vaccination programs. And the Nigerian boycott is an excellent example of that. So let's take a look at what happened there. Okay, so public trust is essential in promoting public health. Such trust plays an important role in the public's compliance with public health interventions, especially compliance with vaccination programs, which target mainly healthy people. Where public trust is eroded, rumors can spread, and this can lead to rejection of health interventions. In northern Nigeria in 2003, the political and religious leaders of Kano, Zamfara, and Kaduna states brought the immunization campaign to a halt by calling on parents not to allow their children to be immunized. These leaders argued that the vaccine could be contaminated with anti-fertility agents, HIV, and cancerous agents. This impasse was eventually resolved in July 2004 through dialogue with religious leaders playing a significant role in the process. The federal government had invited political and religious leaders to a series of meetings in order to find a solution to the impasse. The WHO and UNICEF also played a role in breaking the deadlock. An agreement was reached to test the vaccine in other independent countries. In February 2004, the Nigerian government sent state and religious representatives to South Africa, Indonesia, and India to observe testing of the polio vaccine and to bring back proof that it was not contaminated with HIV. The vaccine boycott in Nigeria was influenced by a complex interplay of factors. These factors included lack of trust in modern medicine, political and religious motives, a history of perceived betrayal by the federal government, the medical establishment, and big business, and also a attempt by local leadership to protect its people. During the ban, the polio virus spread from Nigeria and reinfected 23 previously polio-free countries around the world, including nations as far away as Indonesia and Yemen, causing nearly 1,500 children to be paralyzed for life. The bottom line for all of this is that immunization campaign programs should be a participatory event involving state and local governments, community leaders, and the participants, oftentimes the parents of the children who are going to receive the vaccines. What we see is that when these efforts were made, it had terrific effects. So after the concerns of local officials regarding the vaccine were assuaged, the boycott was lifted and vaccination programs resumed. On August 25, 2020, after 4 years without a single case of wild polio, the African region has been certified free of wild polio. So decades of extraordinary investment has finally paid off. In 1988, polio was endemic in 125 countries, according to UNICEF. Since then, through aggressive vaccination programs, the virus has been almost eradicated in most countries, with the notable exceptions of Pakistan and Afghanistan. In 2020, there were 73 wild polio cases reported in Pakistan, compared to 147 in all of 2019. In Afghanistan, there were 47 wild polio cases so far this year, compared to a total of only 29 in all of 2019. So there's still virus transmission going on in these countries. So let's move into some aspects of how vaccines work. At the heart of every vaccine is an antigen, so called because it is the thing that provokes the body to generate antibodies, as well as other immune responses. The adaptive immune system possesses a memory component that allows for an efficient and dramatic response upon reinvasion of the same pathogen. Memory is handled by the adaptive immune system with little reliance on cues from the innate response. During the adaptive immune response to a pathogen that has not been encountered before, called a primary response, plasma cells secreting antibodies and differentiated T cells increase, then plateau over time. As B and T cells mature into effector cells, a subset of the naive populations differentiate into B and T memory cells with the same antigen specificities. A memory cell is an antigen-specific B or T lymphocyte that does not differentiate into effector cells during the primary immune response, but that can immediately become effector cells upon re-exposure to the same pathogen. During the primary immune response, memory cells do not respond to antigens and do not contribute to host defenses. As the infection is cleared and pathogenic stimuli subside, the effectors are no longer needed, and they undergo apoptosis. In contrast, the memory cells persist in the circulation. If the pathogen is never encountered again during the individual's lifetime, B and T memory cells will circulate for a few years or even several decades, but will eventually die off, having never functioned as effector cells. However, if the host is re-exposed to the same pathogen type, circulating memory cells will immediately differentiate into plasma cells and CTLs without input from the APCs or the T cells. One reason the adaptive immune response is delayed is because it takes time for naive B and T cells with the appropriate antigen specificities to be identified and activated. Upon reinfection, this step is skipped, and the result is more rapid production of immune defenses. Memory B cells that differentiate into plasma cells output tens to hundredfold greater antibody amounts than were secreted during the primary response. The rapid and dramatic antibody response may stop the infection before it can even become established, and the individual may not realize they had even been exposed. Vaccination is based on the knowledge that exposure to non-infectious antigens derived from known pathogens generates a mild primary immune response. The immune response to vaccination may not be perceived by the host as illness, but still confers immune memory. When exposed to the corresponding pathogen to which an individual was vaccinated, the reaction is similar to a secondary exposure. Because each reinfection generates more memory cells and increased resistance to the pathogen, and because some memory cells die, certain vaccine courses involve one or more booster vaccinations to mimic repeat exposures. For instance, with tetanus, there's a very effective vaccine for that. Now, I know tetanus is not a vaccine, it's not a virus, but you need to get a tetanus booster every 10 years because that's how long the memory cells last that are directed against tetanus. Okay, now we want to go on to discuss different types of vaccines. And there are many different kinds that we can talk about. But originally, during the development of vaccines, two types were the most common: the live attenuated virus vaccines and the inactivated or killed virus vaccines. Live vaccines are derived from wild or disease-causing pathogens. These wild viruses are attenuated or weakened in a laboratory, usually by repeated culturing. For example, the measles virus used as vaccine today was isolated from a child with measles disease in 1954. Almost 10 years of serial passage using tissue culture media was required to transform the wild virus into attenuated vaccine virus. To produce an immune response, LAVs must replicate, that is, grow, in the vaccinated person. A relatively small dose is administered, which replicates in the body and creates enough of the virus to stimulate immune responses. Although LAVs replicate, they usually do not cause disease, such as may occur with the wild form of the virus. When a live attenuated vaccine does cause disease, it's usually much milder than the natural disease and is referred to as an adverse reaction. The immune response to a live attenuated vaccine is virtually identical to that produced by a natural infection. The immune system does not differentiate between an infection with a weakened vaccine virus and an infection with a wild virus. Live attenuated vaccines produce immunity in most recipients with one dose, except those administered orally, such as the oral polio vaccine. However, a small percentage of recipients do not respond to the first dose of an injected live vaccine, such as the MMR or varicella vaccines, and a second dose is recommended to provide a very high level of immunity in the population. Live attenuated vaccines may cause severe or fatal reactions as a result of uncontrolled replication of the virus vaccine. This only occurs in persons with immunodeficiency, such as from leukemia, or treatment with certain drugs, or HIV infection. A live attenuated vaccine virus could theoretically revert to its original pathogenic form. This is known to happen only with the live oral polio vaccine, and we'll talk about that in a minute. Active immunity from a live attenuated vaccine may not develop because of interference from circulating antibody to the vaccine virus. Antibody from any source, such as transplacental or transfusion, can interfere with replication of the vaccine virus and lead to poor response or no response to the vaccine. This is called vaccine failure. We can see vaccine failure in puppies given the parvovirus vaccine due to maternal antibodies. To get around this, we vaccinate puppies in a series, giving the vaccine every 2 to 4 weeks until at least age 16 weeks and sometimes 20 weeks. By giving the vaccine at intervals, we're trying to catch the puppy's immune system after their window of vulnerability is closed. We know that by age 16 weeks, we should be able to get a vaccine to take, but we still begin at a younger age in an effort to provide immunization as soon as possible, especially as some of the pups won't have an appropriate maternal antibody response. Live attenuated vaccines are fragile and can be damaged or destroyed by heat and light. They must be handled and stored carefully. Currently available live attenuated viral vaccines are measles, mumps, rubella, vaccinia, varicella, zoster, which contains the same virus as the varicella vaccine but in a much higher amount, yellow fever vaccine, rotavirus, and the intranasal influenza vaccine. The oral polio vaccine is a live viral vaccine, but it's no longer available in the United States, and we'll talk about why again in a few minutes. For inactivated viruses, the specific virus is killed with heat or chemicals, and its dead cells, or virions, are introduced into the body. Even though the pathogen is dead, the immune system can still learn from its antigens how to fight live versions of it in the future. These viruses can be freeze-dried and easily stored because there's no risk of killing the pathogen, like there is with live attenuated vaccines. They're also safer without the risk of the virus mutating back to its disease-causing form. However, because the virus is dead, it's not as immunologically stimulating as the original live virus. And it's not as immunologically stimulating as a live attenuated vaccine virus. Therefore, it often takes several doses and booster shots to build up sufficiently robust immunity. Some of the vaccines that are inactivated virus vaccines would include the IPV polio vaccine, hepatitis A, and the human rabies vaccine. Inactivated vaccines always require multiple doses. In general, the first dose does not produce protective immunity, but rather primes the immune system. A protective immune response develops after the second or even after the third dose. In contrast to live vaccines, in which the immune response closely resembles natural infection, the immune response to an inactivated vaccine is mostly humoral. Little or no cellular immunity results. Antibody titers against inactivated antigens diminish with time, and as a result, some inactivated vaccines may require periodic supplemental doses to increase or boost antibody titers. As I previously mentioned, attenuated vaccine viruses can potentially revert to their original disease-causing form. This is currently known only to happen with the live oral polio vaccine. So let's take a look at a comparison between the live and attenuated polio vaccines. So as I mentioned, there are two vaccines for polio, the oral polio vaccine, or OPV, and the inactivated polio vaccine, or IPV. OPV is taken orally as drops and can be easily administered. It does not require a trained health worker. OPV is still the main protective measure against polio. IPV is given through injection by a trained health worker. In countries still using OPV, or the live virus vaccine, IPV does not replace the OPV vaccine, but is used with the OPV to strengthen a child's immune system and to protect them from polio. Until polio is eradicated globally, OPV is still the main preventative measure used against polio. IPV is recommended in addition to OPV and does not replace OPV. Vaccine-derived polioviruses, or VDPVs, are rare strains of poliovirus that have genetically mutated from the strain contained in the oral polio vaccine. The oral vaccine contains a live attenuated vaccine virus. When a child is vaccinated, the weakened vaccine virus replicates in the intestine and enters into the bloodstream, triggering a protective immune response in the child. Like wild poliovirus, the child excretes the vaccine virus for a period of 6 to 8 weeks. Importantly, as it's excreted, some of the vaccine virus may no longer be the same as the original vaccine virus because it's become genetically altered. In other words, it's mutated during its replication in the child's gut. OPV is very effective against the wild poliovirus, but in very rare cases, the vaccine can lead to paralysis. One type of polio caused by OPV is called vaccine-associated paralytic poliomyelitis, or VAP. About 40% of these cases are caused by the OPV's type 2 component. There are 3 components to a polio vaccine because there are 3 types of polio. And it's primarily the type 2 in the oral vaccine that causes this paralysis. Another form of vaccine-associated polio is the circulating vaccine-derived polio, or CVDPV. These are mutated versions of OPV which can cause paralysis and spread from person to person. Almost all of these outbreaks in recent years have been caused by type 2 vaccine-derived virus. Now, the wild polio virus type 2 was eradicated globally in 1999. Since the risk of paralytic disease due to the type 2 virus in the OPV and that risk outweighs the benefits, the trivalent OPV vaccine that has previously been used is being replaced with the bivalent oral vaccine. The bivalent will continue to target the remaining polio types, types 1 and 3. The switch from trivalent OPV to bivalent OPV will significantly reduce the risk of VAP and CVDPV. Increased protection from types 1 and 3 virus with the bivalent OPV will still be a major benefit to this, because the bivalent OPV is a more powerful vaccine than the trivalent OPV was against these types of viruses. Therefore, the switch from the trivalent OPV to the bivalent OPV will offer increased immunity to the remaining types of wild polio virus. Once types 1 and 3 are eradicated, they'll stop using the oral vaccine altogether. And that's what we do here in the U.S. We have eliminated wild polio from the U.S., and so we no longer use the oral vaccine because it's not worth the risk. The immunity that you develop from the killed virus vaccine isn't quite as robust, and it requires that you get your vaccine from a trained medical person because it requires an injection, and those kinds of issues still remain, but that's not a major impediment to us here in the U.S. So we have stopped using the oral polio vaccine. Although, sadly to admit, I'm old enough to remember when they started the big vaccination program with the OPV. It was a community event. Entire families went together into gyms and churches and so on to get this vaccine. And it was given to you in just a little cup in a little bit of sugar water, because remember, the OPV is taken orally. And it was such a huge deal because the concerns of paralytic polio breaking out in the population every summer was, it was a very serious threat, particularly to children. Okay, so now we want to move on to talk about some of the new vaccine technologies. And there are many different kinds of methods and platforms that are being developed. There's subunit vaccines, DNA vaccines, even RNA vaccines. We'll talk about that at the end of the lecture when we look at coronavirus vaccines. There are virus-like particles and live recombinant vaccines. So let's go through some of these. With a subunit vaccine, instead of the entire virus, it includes only the antigens that best stimulate the immune system. In some cases, these vaccines use epitopes, the very specific parts of the antigen that antibodies or T cells recognize and bind to. Because subunit vaccines contain only the essential antigens and not all the other parts of the virus, the chances of adverse reactions to the vaccine are lower. Subunit vaccines can contain anywhere from 1 to 20 or more antigens. Of course, identifying which antigens best stimulate the immune system is a tricky, time-consuming process. Once scientists do that, however, they can make subunit vaccines in one of two ways. Then they can either grow the virus in the laboratory and then use chemicals to break it apart and gather the important antigens, or they can manufacture the antigen molecules from the virus using recombinant DNA technology. Vaccines produced in this way are called recombinant subunit vaccines. Advantages to this would include there's no contamination from the original virus. You can avoid issues like we see with egg allergies for some vaccines that are produced in chicken eggs, because you can produce your vaccine in things such as E. coli or yeast. However, many of these vaccines will fail because they're insufficiently immunogenic. Now, hepatitis B is a success story. It uses the viral surface antigen in that particular vaccine. The hepatitis B vaccine is a subunit vaccine, and it's very effective. The surface antigen to the virus is cloned and inserted into yeast cells, just common baker's yeast. The yeast then produces the antigen, which is then collected and purified for use in the vaccine. Very easy procedure, much easier to produce than using cell culture or eggs and some of these other methods that are used for vaccine production. They're continuing research to develop a recombinant subunit vaccine against hepatitis C virus. With human papillomavirus vaccines, they're built on a virus-like particle platform. And there are two different companies that are producing papillomavirus vaccines. Both of them are using this VLP approach. And the vaccines are composed of a single protein, L1, which is the major structural capsid protein of the virus, and it contains the immunodominant neutralization epitopes of the virus. The vaccines are based primarily on preclinical research showing that when expressed in cells, L1 has the extrinsic ability to self-assemble into VLPs, or virus-like particles, that can induce high levels of neutralizing antibodies. Both vaccines target HPV-16 and HPV-18, which account for approximately 70% of cervical cancers. The Merck vaccine also targets HPV-6 and HPV-11, which account for approximately 90% of external genital warts. Although still in the experimental stages, DNA vaccines show great promise, and several types are currently being tested in humans. DNA vaccines take immunization to a new technological level. These vaccines dispense with both the whole organism and its parts and get right down to the essentials, the virus's genetic material. In particular, DNA vaccines use the genes that code for those all-important antigens. Researchers have found that when the genes for a virus's antigens are introduced into the body, some cells will take up that DNA. The DNA then instructs those cells to make the antigen molecule. The cells secrete the antigens and display them on their surfaces. In other words, the body's own cells become vaccine-making factories, creating the antigens necessary to stimulate the immune system. A DNA vaccine against a virus would evoke a strong antibody response to the free-floating antigen secreted by the cells, and the vaccine would also stimulate a strong cellular response against the viral antigens displayed on cell surfaces. The DNA vaccine couldn't cause a disease because it wouldn't contain all of the genes of the virus, just copies of a few of the genes. In addition, DNA vaccines are relatively easy and inexpensive to design and produce. So-called naked DNA vaccines consist of DNA that is administered directly into the body. These vaccines can be administered with a needle and syringe or with a needleless device that uses high-pressure gas to shoot microscopic gold particles coated with DNA directly into the cells. Sometimes the DNA is mixed with molecules that facilitate its uptake by the body's cells. Naked DNA vaccines being tested in humans include those against viruses that cause influenza and herpes. Here's an example of a DNA vaccine that's been developed for use against Zika virus. For a DNA vaccine against Zika, scientists inserted genes for the Zika proteins into a circular piece of DNA, the plasmid, and many copies of the plasmid are injected into muscle. Some of the DNA travels into the cell's nuclei, where it is used to make messenger RNA. After exiting the nucleus, the mRNA helps build Zika proteins that can then form virus-like particles that trigger the immune system to make antibodies. There are a couple of Zika virus DNA vaccines that are in phase 1 clinical trials right now. To finish up this lecture, let's look at a promising new coronavirus vaccine candidate. It's a very innovative platform, and this vaccine is being produced by Moderna Therapeutics. They just recently moved into phase 3 of clinical trials with this vaccine. Their phase 3 trial will include an estimated 30,000 participants tested at 89 sites spread across 30 states and the District of Columbia. The aim is to determine if 2 doses of this mRNA vaccine will prevent people from catching COVID-19, stop those with the disease from dying, or both. mRNA vaccines take a novel approach that's never been licensed for human use. Their development is faster as it bypasses the more laborious tasks of inactivating viruses or isolating proteins. Essentially, the vaccine is composed of messenger RNA for the virus spike protein. These messenger RNAs have been put into a lipid nanoparticle for delivery into the cell. Once the messenger RNA is delivered into the cell's ribosomes will start translating the messenger RNA to form spike proteins. These spike proteins, in turn, will trigger an immune response. So it's a pretty cool system. We'll have to see how it works out. Recombinant vector vaccines are experimental vaccines similar to DNA viruses, but they use an attenuated virus or bacterium to introduce the viral DNA to cells of the body. Vector refers to the virus or bacterium used as the carrier. In nature, viruses latch onto cells and inject their genetic material into them. In the lab, scientists have taken advantage of this process. They have figured out how to take the roomy genomes of certain harmless or attenuated viruses and insert portions of the genetic material from other viruses into them. The carrier viruses then carry that viral DNA to cells. Recombinant vector vaccines closely mimic a natural infection and therefore do a good job of stimulating the immune system. Researchers are working on both bacterial and viral-based recombinant vector vaccines for HIV, rabies, and measles. An example of a recombinant vaccine currently in use is the VRG vaccine for rabies. Now, this is only used in animals and not in people. The vaccine was produced by inserting the glycoprotein protein gene from rabies into a vaccinia virus. It is harmless to humans and has been shown to be safe for various species of animals that might accidentally encounter it in the wild, including birds. VRG has been successfully used in the field in Belgium, France, Germany, and the U.S. to prevent outbreaks of rabies in wildlife. The vaccine is stable under relatively high temperatures and can be delivered orally, making mass vaccination of wildlife possible by putting it in baits. The plan for immunization of normal populations involves dropping bait containing food wrapped around a small dose of the live virus. The bait would be dropped by helicopter, concentrating on areas that have not been infected yet. Just such a strategy of oral immunization of foxes in Europe has already achieved substantial reductions in the incidence of human rabies. A strategy of vaccinating neighborhood dogs in parts of India, combined with a sterilization program of the dogs, has also resulted in a large reduction in the number of human cases. The experimental Ebola vaccine currently being used to contain the outbreak in the Democratic Republic of the Congo is a recombinant vaccine using VSV, and it's been shown to be protective 97.5% of the time, according to new data that's been released by the WHO. Although several studies have shown that the vaccine is safe and protective against the Ebola virus, more research is necessary before the vaccine can be licensed. The vaccine is therefore currently being used on a compassionate basis to protect persons at highest risk of Ebola during this outbreak under a ring vaccination strategy, which is similar to the approach that we talked about that was used to eradicate smallpox. The ring is not necessarily a contiguous geographic area, but rather captures a social network of individuals and locations that may include dwellings or workplaces further afield, where the index patient spent time while asymptomatic, or the households of individuals who had contact with the patient during the illness or after his or her death. Experience suggests that each ring may be composed of an average of about 150 people. This figure shows the Northern Hemisphere influenza vaccine production timeline. Global viral surveillance occurs year-round, which informs the selection of the vaccine strains that occurs between January and March of each year. Vaccine production begins in January and continues through July. Purification and testing starts in June and continues through October. Single-dose syringes and multi-dose vial lots are filled and packaged between July and December and are distributed between August and December for vaccination between late September and the end of influenza season. So the whole process of identifying your vaccine strains, making the vaccine, purifying it, packaging it, but let's not forget the distribution efforts as well. It's a very complicated process, and it has to work along the appropriate timeline, because if you deliver your vaccine at the wrong time of year, it's going to be worthless. This figure shows the structure and replication cycle of influenza A virus and some of the characterized adaptive immune responses. There are 8 genome segments that code for 11 proteins. That includes the hemagglutinin, or the HA, and the neuraminidase, the NA, which account for most of the known antigenic determinants. The portion of the matrix protein, M2, that is outside of the viral envelope is also antigenic. Adaptive immune responses are shown in this figure in panels A, B, C, and D. Now take a look at panel A. With that, you see that the influenza HA protein mediates attachment of the virus to its host cell receptor. Antibodies directed against the HA protein block attachment of the virus to the host cell receptor or block fusion of the virus and host membrane. Antibodies generated against HA are correlated with vaccine protection against infection. If you look at panel B, you see antibodies generated against the NA protein do not protect infection, but limit the release of virus from infected cells. Antibodies directed against NA have been correlated with a reduction in disease severity. In panel C, antibodies generated against the highly conserved external domain of the M2 protein interfere with virus assembly or constrain proton transport and are highly cross-reactive across virus subtypes. In panel D, CD8 T-cell responses to conserved influenza virus components have been correlated with enhanced clearance of virally infected cells. However, the exact degree to which they contribute to a reduction in illness remains uncertain. Here you see several of the new approaches that have been developed to trying to come up with new influenza vaccines. In A, you can see that the HA gene is cloned into a vector which expresses the recombinant HA protein in infected cells, and the protein is subsequently purified. In panel B, the simultaneous infection of cells with individual vectors containing the HA, NA, and M1 genes respectively results in self-assembling virus-like particles that contain the HA and NA proteins on their surface, but lack the influenza gene segments, so they're non-replicating. In panel C, the influenza HA protein is expressed by the surface of a carrier virus that presents it to the immune system, but that cannot itself cause disease. And in D, a DNA vaccine consists of a DNA plasmid into which one or more influenza virus genes are inserted. Okay, now we need to talk about adjuvants and vaccine delivery systems. Aside from antigens, ingredient components of a vaccine include adjuvants, which are added to enhance the immune system response, antibiotics to prevent contamination during the manufacturing process, and preservatives and stabilizers. Adjuvants especially stimulate the inflammatory response. They mimic or induce cellular damage, stress, and lead to the release of heat shock proteins, which are danger signals. One commonly used adjuvant would be Freund's, which is killed mycobacteria suspended in mineral oil. So adjuvants play an important role, particularly for a killed virus vaccine. And then, of course, we've already talked to some extent about vaccine delivery systems, because remember, the oral polio vaccine is just taken as a liquid drop. Of course, there are hypodermics used to deliver a lot of vaccines. And we mentioned the gene guns, the air guns, and so on. But another type of delivery system that's being looked at is edible plants. They're producing vaccines in transgenic plants, which could reduce the cost of production and reduces the need for cold storage. And of course, you don't need needles. And they refer to this as Farmageddon. But edible plants are very effective as a delivery vehicle for inducing oral immunization. So, of course, your vaccine is going to have to be able to be taken orally. If it's inactivated in the gut, then you can't use an edible vaccine. But for instance, polio, which goes through the digestive system during a normal infection, can be used in a system like this. And of course, with an edible plant, you're not going to need an adjuvant, and it's much easier to administer than an injection. Ask a kid to eat a banana rather than roll up their sleeve and get a shot. I think you can figure out for most kids they'd rather eat the banana. It's easy for separation and purification of vaccines from plant materials, and it's convenient. There's safety in storing and transporting vaccines this way. So it's a pretty good system, but it does have some limitations. For instance, producing stable and reliable amounts of vaccines in plants is complicated by the fact that tomatoes and bananas don't come in standard sizes. So, you have to worry about, could there be a situation where people could ingest too much of the vaccine, which could be toxic, or too little, which could lead to disease outbreaks among populations believed to be immune. So there's still problems that have to be worked out, but it's really an interesting approach. Now, when most people hear the word vaccine, they think of a way to prevent disease. However, therapeutic vaccines aren't used for prevention. Instead, they're used as a method of treatment. Just like a regular vaccine, therapeutic vaccines are used to stimulate the immune system to target an infection or a type of disease cell, such as a cancer cell. In other words, therapeutic vaccines help teach the body how to do a better job of protecting itself. The goal is to boost the immune system to help it control or get rid of otherwise difficult to treat conditions. One example of a therapeutic vaccine is Shingrix, the shingles vaccine. Shingles results from the reactivation of varicella zoster virus in a patient with a history of varicella or chickenpox infection. Shingrix works to boost the body's protection against shingles and is recommended for those over 50. Thimerosal is a mercury-containing preservative used in some vaccines and other products since the 1930s. There's no convincing scientific evidence of harm caused by the low doses of thimerosal in vaccines, except for minor reactions like redness and swelling at the injection site. However, in July 1999, the Public Health Service agencies, the American Academy of Pediatrics, and vaccine manufacturers agreed that thimerosal should be reduced or eliminated in vaccines as a precautionary measure. Since 2001, with the exception of some influenza vaccines, thimerosal is not used as a preservative in the routinely recommended childhood vaccines. So, for instance, the MMR vaccine hasn't had thimerosal in it since 2001. Recent estimates from CDC's Autism and Development Disabilities Monitoring Network found that about 1 in 150 children have an ASD. This estimate is higher than estimates from the early 1990s. Some people believe increased exposure to thimerosal from the addition of it to all the new vaccines that are now recommended for children. They think that this explains the higher prevalence in recent years. However, evidence from several studies examining trends in vaccine use and changes in autism frequency really does not support such an association. I know that there's some strongly held beliefs about this, and all I can do is encourage you to, read the literature, and in particular, read the criticisms of the original paper that was published in The Lancet that started this whole controversy about MMR and autism. Researchers have been working on an HIV vaccine since the 1980s, but progress towards an effective vaccine has been much slower than anticipated. Most vaccines against other diseases stimulate the production of antibodies that neutralize virus infectivity. But in the case of HIV, neutralizing antibodies do not clear the infection. This is because HIV reproduces so fast and mutates so quickly that antibodies produced against the virus quickly become ineffective against the newer mutated viruses. Millions of new viruses are produced each day, and each one is slightly different from the previous generations of the virus. Antibodies against HIV are only likely to be effective if they can bind to regions of the virus that vary little between these individual virions. Another issue is that HIV has several subtypes which are concentrated in different regions of the world. For example, subtype B is common in North America and Europe, but subtype C is common in Southern and Eastern Africa. Any vaccine must either be effective against all subtypes, or different vaccines must be developed against the various subtypes. A vaccine might also stimulate the production of immune system cells called T lymphocytes that can clear HIV-infected cells. But HIV has also evolved to suppress some immune responses that are important in the early stages of viral infection. Another challenge in vaccine development for HIV is finding efficient ways to deliver HIV proteins safely in ways that will allow the immune system to recognize HIV and respond to it without establishing an infection. Remember, HIV integrates into human cells, remember the provirus, and uses those cells to reproduce. So live or attenuated whole virus vaccines are completely unsuitable for use in HIV. Instead, HIV proteins need to be engineered in ways that make them harmless, but still recognizable to the immune system. These proteins or sequences of viral material must be delivered using a vector, another harmless virus such as canarypox or adenovirus, a common cold virus, which presents them to the immune system. Now you can see that there are a lot of different types of vaccines in development for HIV, and they're in all different stages of trials right now. So there's a lot of effort and energy being directed against this project. The RV144 study in Thailand was a landmark because it was the first, and to date only, large clinical trial to demonstrate efficacy for an investigational HIV vaccine. At the end of the 3.5-year study period, investigators observed a 31% reduction in HIV infection among vaccine recipients compared to those who received a placebo. The trial, which involved more than 16,000 adult participants, was sponsored by the U.S. military in collaboration with NIAID and other partners. RV144 evaluated the safety and effectiveness of a prime boost combination of two vaccine components given in sequence. So not just one vaccine, but two. There was the Alvac HIV, which uses canarypox virus as a vector to deliver HIV genes, and the AIDS Vax BE, which contains a protein found on the HIV surface. The vaccine components, which individually had not protected recipients against HIV infection, were based on the HIV B and E subtypes common in Thailand. This prime boost regimen appeared to provide the greatest protective effect during the first year after vaccination, providing a 60% reduction in infection risk, but the level of protection decreased over time. The HVTN 702 study was designed to build on the positive results that were found with the RV144 trial in Thailand. In this trial, it began in 2016. It was taking place in South Africa, and it was testing an investigational prime boost vaccine similar to the one used in Thailand. And it was somewhat different because it was designed to work against the clade C HIV subtype, which is most common in Africa, where the pandemic is most pervasive. This study enrolled 5,400 HIV-negative volunteers at 14 sites across South Africa. The study population consisted of sexually active men and women aged 18 to 35 years. The study volunteers were randomly assigned to receive either the investigational vaccine regimen or a placebo. Study participants received 6 injections over 18 months. However, in January of 2020, just a few months ago, during an interim analysis, they found that although the virus was safe, it was not effective, and they canceled the program. What they had found is that after 18 months, when they looked at HIV infections in the groups, they found 129 HIV infections in the vaccine recipients and 123 HIV infections in the placebo recipients. So they decided based on these findings that the vaccine had not shown any efficacy. So they stopped the program. No further vaccinations were administered, but they continue a follow-up study of all the participants. But as you can imagine, this was a major disappointment. A current study going on is the Imbokodo study. It's also testing a prime boost vaccine, a somewhat different design than the one that was used in the previous 2 trials that we talked about. This one combines a prime vaccine that uses an adenovirus vector to deliver a mosaic protein designed to produce responses against a wide range of viruses, and an HIV subtype C booster designed to stimulate the production of antibodies against the HIV envelope protein GP140. The vaccine is being tested on 2,600 women aged 18 to 35 in southern and eastern Africa. The results are expected by 2020. But this is a tetravalent vaccine, and like I said, it contains a mosaic of HIV genes from different subtypes of the virus that have been stitched into a harmless adenovirus. In earlier trials, this vaccine did not trigger neutralizing antibodies, but produced high levels of binding antibodies and other immune responses. A booster shot consists of a mosaic version of HIV's surface protein, the GP140, mixed with an alum adjuvant. And so right now, this is in phase 2B proof of concept efficacy studies. And as I said, it'll be 2022 before they have the results, but they've successfully enrolled 2,600 participants. Now, in this particular study, they're using women. Women are in the study are all of the age who would be at risk of acquiring HIV. Another study that's going on is the Mosaico study. This is in US, Latin America, and Europe. This will test a vaccine that is very similar to the one being tested in the African women's trial, but it will be studied in other parts of the world, as I said, the US, Latin America, and Europe. It will be provided to members of communities where the risk of HIV is also high, among people who are transgender and men who have sex with men. This is in contrast to Africa, where women bear the brunt of new infections. So that's why women were chosen for the African study. And here in this Mosaico study, they're using men. So in the US and Europe, men who have sex with men account for 57% of new HIV infections. Also at higher risk of HIV are transgender people. In a survey of 3 million HIV test results reported to the CDC, the percentage of transgender people who newly acquired HIV was triple that of the US national average. And of course, think about it, when you're trying to do a vaccine trial, you have to be trying it on populations that are at highest risk of exposure to the virus. The HIV-negative volunteers, just as in the Mbokodo study, will receive 4 injections over the course of the year. Some people will get the vaccine, some will get the placebo. And it's a mosaic design for this particular vaccine as well. Now, there are therapeutic vaccines that are being looked at for HIV. So remember, a therapeutic vaccine is designed to improve the body's immune response. In this case, the body's immune response to HIV in a person who's already infected with HIV. Researchers are developing and testing therapeutic HIV vaccines to slow down the progression of HIV to AIDS, and treating people with these vaccines would help keep HIV at undetectable levels without the need for regular antiretroviral therapy, which is expensive and also has significant side effects to the recipient. Currently, a person with HIV must remain on antiretroviral therapy to keep their HIV virus levels at undetectable levels. A therapeutic HIV vaccine may also make it less likely that a person could transmit HIV to others. Currently, there's no therapeutic HIV vaccine that's been approved by the FDA, but research is underway, and it could be possible to get enrolled in a clinical trial to receive one of these therapeutic HIV vaccines. So as I mentioned before, researchers have been working on an HIV vaccine since the 1980s, when we first recognized the HIV virus. But progress has been very slow. Finding at least a partially effective vaccine remains of critical importance for the HIV response. The biggest reduction in new infections would be achieved by a combination of PrEP, which is a pre-exposure prophylaxis that's used when people are at risk for HIV. They'll take a daily medication to prevent getting HIV. So PrEP can stop HIV from taking hold and spreading. Then we have antiretroviral treatments for people who are already infected with HIV, and then if we had a vaccine. The 3 of these things together could go a long way to really bringing the numbers of HIV infections down. So to still be useful, an HIV vaccine doesn't have to have the 90% or 99% efficacy that we saw with things like smallpox and polio. An HIV vaccine is a more realistic prospect today than a decade ago, and an optimistic forecast of HIV vaccine availability is that one might be available by 2030. Now, the topic of vaccine safety became prominent during the mid-1970s, with increases in lawsuits filed on behalf of those that had presumably been injured by the diphtheria, pertussis, and tetanus, or DPT vaccine. Legal decisions were made and damages awarded despite the lack of scientific evidence to support vaccine injury claims. As a result of the liability, prices on vaccines of all types soared, and several manufacturers just went out of the business of vaccine production. A vaccine shortage resulted, and public health officials became concerned about the return of epidemic disease. In order to reduce liability and respond to public health concerns, Congress passed the National Childhood Vaccine Injury Act in 1986. This act was influential in many ways. For one thing, it mandated that all healthcare providers report certain adverse events that occur following a vaccination. As a result, the Vaccine Adverse Events Reporting System, or VAERS, was established by the FDA and the CDC. VAERS provides a mechanism for the collection and analysis of adverse events associated with vaccines currently licensed in the United States. Adverse events are defined as health effects that occur after immunization that may or may not be related to the vaccine itself. VAERS data are continually monitored in order to detect previously unknown adverse events or increases in known adverse events. Now, adverse events can be anything as minor as fainting when you get your vaccine shot to something as severe as paralysis, like we saw with polio. So, the National Childhood Vaccine Injury Act of 1986 was established. In addition to VAERS, it also established the National Vaccine Injury Compensation Program. This program is intended to compensate those individuals who have been injured by vaccines on a no-fault basis. No fault means that people filing claims are not required to prove negligence on the part of either the healthcare provider or manufacturer to receive compensation. The program covers all routinely recommended childhood vaccinations. Settlements are based on the vaccine injury table, which summarizes the adverse events caused by various vaccines. This table was developed by a panel of experts who reviewed the medical literature and identified serious adverse events that are reasonably certain to be caused by vaccines. Examples of table injuries include anaphylaxis, or severe allergic reaction, paralytic polio, or encephalopathy. The vaccine injury table was created to justly compensate those injured by vaccines while separating out unrelated claims. As more information becomes available from research on vaccine side effects, the vaccine injury table is updated. But this is to prevent the need for someone who feels that they or their child's been injured by a vaccine needing to go out, get attorneys, and sue a big pharmaceutical company.